talking in the beginning, and then also a majority of all the work was being conducted within the Hakim's lab that was located within to the city region. So traveling from Pinyon to the city location constantly, weekly, but we managed to get the work done. Thank you very much. And also wanted to throw in, my name is Ian Nez. My clans are Tapan Shlento, Tosom Libati, Tachini Dashido, Tanazani Dashnale, Le Beke Pahoza de Nasha. Alrighty, let me start off with the. This is the beginning of my project. Like I said, was the title evaluating the existence of Helicobacter pylori, unique markers of tag A and BAC A genes, and the water samples collected from the Navajo Nation. Next slide. I don't think it's moving. I think it's frozen.
It is taking time on the screen here. From here, it is gone.
Oh, okay. Sorry, we're just having a little bit of difficulties with the technology.
Yeah, it is moving from here.
Is the screen?
Yeah.
Oh, it's the screen.
Just please. Yeah, I have shared. Let me share it again. Sorry for because we are sharing it and it should be.
Okay, this will be the table of contents upon the main points that I'll be touching upon within this thesis defense. Next slide, please.
It is Zoom that is having some issue.
Okay. Sorry, it's our, most likely our side. Let's see. All right. I would like to point out that I received in the beginning of the project, when informing upon the purpose of the project, we got a letter for our DC IRB for an exemption letter that pretty much informs us that while working on this project, it involves no human contact or animal life that will be harmed, but mainly focusing upon the bacterium itself from the collected water samples. Multiple clinical and public health studies were state that roughly 56 to 68% of the Navajo adults tested positive for Helicobacter pylori infection. I'd like to point out that below is will be the gram-negative bacterium with Helicobacter pylori that has a pretty much a bird morphology, and also shows the spiral morphology that's being displayed within these gram-negatives that result within a pinkish coloring. And sorry, I would like to also point out that this Helicobacter pylori is also associated with gastritis and also peptic ulcers, and also leads to stomach cancer. So the question comes from where are we contracting this bacterium, and is it spreading from unregulated water resources in the region? Helicobacter pylori is a gram-negative bacterium that is primarily contracted in childhood, most likely during the first years of life. Through the contaminated water, and older adults have encountered it during their childhood. This infection is one of the most prevalent chronic bacterial infections worldwide, affecting more than 50% of the global population. The prevalence of this rate varies significantly by geological regions and are strongly influenced by sociological, environmental, and hygiene-related factors. The highest infection rates are reported in developing countries where limited access to water, clean water, inadequate sanitation, and crowded living conditions contribute to the increased transmission. So most likely the living status that resides within the Navajo reservation does contribute towards these type of conditions. The indigenous communities in the United States have elevated rates of infection and gastric cancers. Members of the Navajo Nation have rates of infection rates between 56 to 70%, and gastric rates between 3 to 4 times higher than the non-Hispanic white populations. Here is a diagram that we are displaying that shown in red are the majority of the prevalence of this Helicobacter pylori within across the world. And then located within the United States had less than 40% of this prevalence of Helicobacter pylori within our region. On the left is a map that we have from the Navajo Partition Land Management units of the windmills that we obtained these water samples from, showing starting from Pinion, Blue Gap, Whipplewell, Low Mountain, Forest Lake, Black Mesa, Chinchipito, Tonalea, Jedito, White Cone, and of course Tuba City region. On the right is a map of all the locations of the windmill that tested positive for DNA bacteriums within our contaminated water samples that we will be displaying within the results. Here is our possible ways of the transmission of how to contract this bacterium. First, starting off within the intrafamilial transmission that starts from either the mother contracting this disease from an adulthood and then passing it on to their children. And then within the family, since close-to-close contact is being possible within each household of the Navajo reservation, this transmission of this bacterium can be passed to one another. And part two is to be the external familial transmission, which is from one household to the next. Since in the majority of the Navajo reservation, majority of the households are close to one another's within the family region. Three is to be our waterborne transmission and how these bacteria, we will be contracting these bacteriums through contaminated food and water, can be found between the fecal matter that's being released between these families and also livestock within the unregulated resources that are being passed through into the unregulated water resources that would be consumed by us and then also be used for our crops that would be consumed rawly that would lead to signs of infection for the Helicobacter pylori. Also, this type of transmission will also be passed on to our livestock, not just our crops, but also our livestock that can lead to transmission of signs of this bacterium for infection. And since majority of the livestock, due to limited resources that are provided on the Navajo reservation, traveling to supermarkets and also obtaining water is very limited and leads towards these unregulated water resources, such as on the right is Peabody Mine, located in Black Mesa, that's ran annually. It will also be used for our livestock resources and our crops. And we will also be taking from these animals, such as using them for our meat and also dairy products that would also have this bacterium. Helicobacter pylori diagnosis procedures, we will have a non-invasive and an invasive test. And in non-invasive, we are not taking from the body, such as using a syringe to take blood. But an invasive would be that we'll be taking samples from the human body, such as blood samples or even fresh tissue samples. In our non-invasive tests, we are using a urea breath test. That's in this type of procedure will be the consumption of a urea solution-based solution that we consume, and if the bacterium is within our stomach, we'll be activating the ammonium and carbon dioxide that will be released and been found within the breath test. Another will be our stool antigen test that we will be analyzing this type of bacterium within our stool. Within our invasive test through patients that have this gastric bacterium through ulcers or even the gastric cancers, we would be looking through and analyzing through biopsies or even through fresh tissue samples. This would be a description of how this bacterium would be traveling and making its progression within our body. Helicobacter pylori is a spiral bacterium that acts as a drill to penetrate and make way through the gastric microbiome. And with the use of the flagella, would navigate through the stomach within the gastric system to pass through the mucous membrane in order to reach the cell lining. But this type of bacterium will have various enzymes that aid within its progression, such as the urease test enzyme, urease enzyme, that basically would be used to neutralize the pH since the stomach will be in an acidic environment. Will change this acidic environment from acidic to alkaline base for the progression of this bacterium that will basically use the adhesive in order to attach onto the cell lining and form lesions and also ulcers that will also release ammonium and carbon dioxide within the gastric epithelium. I would also like to point out that with the genomes that this bacterium has, such as the BAB-A and SAB-A, contributes towards the adhesion that's being used within the outer membrane. And then also the KEG-A and BAC-A genes that will be used to defend against the antibiotics that's being released from the human body, from the human immune system. Here is an experimental flowchart of my water testing and H. pylori analysis for my project, which is basically taking water samples from our water reservoirs and then taking them back to our lab for testing such upon of pH, color, turbidity, odor, and hardness, and then leading to our chemical testing that tests for chloride, iron, ammonia, nitrate, and then even phosphate. While taking these water samples, we will be conducting through IDEX that tests between Colli alert, Pseudo alert, Entra alert, Legi alert that tests for coliforms, Pseudomonas, Enterococci, and Legionella. While taking these water samples will be cultivated on a bacterial media, and then taking these samples and then running them through a DNA extraction to be run onto our Gridion for read sequencing. The methods that we were using within this project would rely upon bacterial culture. Our water samples were inoculated on the Colombian agar, supplemented within the 7% defibrillated sheep split of trimethoprim, bactomycin, polymyxin B, and amphotericin, antibiotics to increase in selectivity of this Helicobacter pylori. All the plates were incubated at a 37 degrees Celsius for 3 to 5 days under a microaerophilic conditions that means between 5 to 6% oxygen and then 10% carbon dioxide within this carbon dioxide incubator. The isolates were being identified on the Colli morphology and positive oxidase and urease and catalyst tests. The reference strain of this Helicobacter pylori, NCTC 11638, was included as a control, but none of our samples, I would like to point out, came positive for Helicobacter pylori. Due to the environmental conditions that this bacteria has moved to convert to variable non-culturable state, which means that even though that this bacterium was alive, it could not be displayed within our media. This is pretty much a flow chart that I would like to point out upon our IDEX quantity trade, where we were able to obtain 100 microliters of our water samples, mix it with our medias, and then put it within our quantity trays, and then for them to be sealed and then to be incubated at a 37 degrees for 24 hours. This is a pretty good description of how our results would come out. On the far left is a colorless results that would result in negatives. On the second one that shows only 2 large positive, which show the change in color that will be measured between the large and then the smaller wells that we would have to calculate through the manufacturer's recommendations. And next to it would be the all cells positive, would be the change within colonies that are being formed within our IDEX trace. And to further investigate, we would use a UV light that would show the growing colonies that we can calculate for positive results. Here is our PCR amplification that we will be using to amplify our DNA within our sample size. On the far left is our flow sheet that we would use for our PCR and PCR to the gene sequencing, in which we would have our sample on the far left that we will have to quantify to 20 nanograms through our nano drop and then run through our PCR process while using our thermal cycler for these PCR samples to be shown visibly on our electrophorescence gel using a 1 kp marker to be documented, and then afterwards ran through Gridion sequencing to be read. And for our Helicobacter pylori, we use the 16S ribosomal RNA for identification. Here on our is basically how we were able to concentrate our water samples. Since water is very diluted, we were able to bring in our water samples, concentrate it as much as possible to form a lysate for lysate preparations with our concentrated water samples for our Helicobacter pylori DNA extraction. Here is another flow sheet of how we ran our DNA extractions, which we have our lysate on the far left. And we ran through two types of washes in order to remove the impurity to get the concentrated DNA sample size. And we even ran our elution buffers twice just to recover as much DNA as possible so that we can run it through our DNA extraction, which is also known as the PureLink Genomic DNA Kit. And then we check the quantifications of our DNA at the end to meet or even have more than 20 nanograms. If we have less than this, then our results would not come out as accurate, or our sorry, it's my brace, or our results will not meet up to the standards. Is it frozen? Yes. Sorry, on our side it's kind of frozen. Yes. It's the screen. Sorry, we're gonna have to restart real quick. All right, on the top left, on our far left side is gonna be our table showings that pretty much show our PCR conditions and the targets that we were aiming for within our reads. And pretty much the genes that were going to be amplified. Then also on the far right will be the primers in use that we adopted these methods from Dr. Munroy's most current paper that he had a collaboration with and his colleagues for 2022, that was published in 2022. On the far right would be the 16S RNA that we would use to target the whole within the Helicobacter pylori in general. On the far left is our PCR conditions that we use for our 16S ribosomal RNA for our amplifications. And this is the flow sheet that we were able to use that shows the denaturization, the annealing, and also the extension within our PCR conditions that breaks the bonds, basically forms with our primers, and then of course forms new polymers at the end for our new strands. Here on our PCR is also another flow sheet that shows the same thing with our KA and our VAC-A genes that were being used within our project for the PCR conditions, for the denaturizations, annealing, and the extensions for both. Next, we moved on to our Gridion-based detection of this Helicobacter pylori DNA. On the far left was our DNA extractions that we got from our water samples that were being concentrated. We add them with our primers for them to be amplified in our PCR and then to be read through our Gridion sequencer. While majority of some people that use the sequencing, they use a Sanger sequencing that would be used for short-term reads, while we were using Gridion that was being used for long-term reads. And the difference between these two processes is that in Sanger, it would normally take about 24 hours for these results to be conducted, but we'll have to follow the traditional of the DNA extraction and then also the amplifications and then falling into the sequencing. While Gridion was more flexible and we don't have to go through this whole process, but we can also skip the amplifications and just go from the DNA extraction straight to the Gridion sequencing. I would like to point out that on the top left would be our Gridion was our flow cell device that shows that there's 5 openings, but on our first we use for our Helicobacter pylori samplings. So they can run multiple of these flow cells at once. That will be one of the pros of this device. The Oxford nanopore platform passes the icon current through the nanopores, measuring the alteration in the current when biological molecules pass through the nanopore or near it. The alteration are documented and translated to identify that the molecule and base modifications, where we will be even able to load up our samples through the spot on sample port cover and be able to run through the channels in each port to end up at the end, but majority of our results will be analyzed through our sensors array. Next will be our results. None of our water samples came positive for the bacterial cultures. And then these are the diagrams from the water analysis from our IDEX quantity trays that I would like to point out, such as locations from, is it putting back up real quick? Such as the locations from Forest Lake, Black Mesa, Blue Gap, and what's this one? I can't really see it. Cool, thank you. Wait, we got one more. Saley and Low Mountain, Whipplewell, and then also, of course, Pinion location, and then our Utah locations, and then Chinchipito, Kianta, Tonalea, Red Lake. One more time, please. And then Ardena-Hutso, and then Hard Rock locations. And then these types of, we'll look at one for now, so we don't go over the time limit. That we were mainly looking, even though we're targeting our Helicobacter pylori as in general, we also wanted to see what else was in the water samples, such as the coliforms that were being in, the Pseudomonas within the water, Enterococci, and Legionella that showed results within several of the wells. So on the bottom left shows that there's about 7 wells that we obtained these samples from. Some did not show any signs of DNA, but some of them even tested positive for within these Collie alert, Pseudo alert, Entro alert, and then Legio alert. On the right, it will be our full summary of all the water samples from the windmills within the Navajo Western and Central Agency, such as in total, we did a sampling of 125 windmills. And within these unregulated water windmills, for 39 tested positive for Collie alert, or coliforms, 51 tested positive for Pseudomonas species, and then 46 tested positive for Enterococci, 22 tested positive for Legionella species. One more, please. Out of all the 125 water samples, only 17 showed somewhat of the DNA presence after PCR. So even after PCR, it does inform us that that the other 108 did not show signs of DNA presence after PCR. Out of the 17 that we tested, only 11 showed the band size close to the Helicobacter pylori base size, and that 11 were ran through our Gridion sequencing to be read, and only 2 of them came up positive for Gridion. Here will be a good example of our electrophorescence gel that the Helicobacter pylori extractions were sample sizes were being taken on July 24, 2025. On the far left will be our 1 kb ladder, and on the far left will be a good legend and also the base number pairs and then the DNA mass. But in our wells 2, 3, and 4 were our Saley Lake, Wheatfields Lake, and Mexican water, in which we obtained that showed DNA presence. For our water samples 1 through 4, DNA extractions were conducted on July 20th of 2025. In this one, I loaded them backwards, but we still had the labeling pretty much done from Chinchipito, Utah, and then also 2 from White Cone within the wells 1, 2, 3, and 4. Within our 5th well will be our 1 kb ladder, in which we also got signs of bacterial presence. And then within our water samples 1 through 10 that was conducted on August 4, 2025, we took 10 samples from White Cone location, and of course we had DNA presence within these 10 samples. And on the far left was our 1 kb ladder. So it was mainly asking the question of which out of these 17 wells had shown signs of DNA of Helicobacter pylori presence. Within our Gridion that we ran for our sequencing from the concentrated water samples from Oxford Nanopore, we did start off with,
He's doing it right now, huh?
pretty much kind of
Yeah.
an old the sample that we collected within the beginning of last year. Then with this, we ran through our Gridion process that shows that within our 48 hours out of the 72 hours that we could ran, we had more fails than pass. So on the bottom left would be the green that indicates the amount of pass, or also signs of DNA being red, and then also the amount of fails. So within our first test, we had more failures due to either it could have been the way our loading was conducted within our Gridion, or it could have been that the results or the sample sizes were past due to
Cool.
the storage that it was kept in, or various other types of technical difficulties that we could have had, such as being an air pocket due
That
to
is super right there.
quality within our Gridion.
Okay.
Therefore, within these, we have to be sure that everything has to be perfect within to meet the qualities for our DNA. Within our second run, that we had more success than failures because we actually ran this process for 24 to 72 hours that gave us more quality and more prolific reads within our process. And of course, it showed more pass than fails. And then of course, for our estimated size of 1.82 gigabytes, we had 1.98 million reds out of 1.26 million fails within our Gridion sequencing runs. Sorry. I would like to inform that within our region bases that must pass a quality score of above 9 in order to pass. So therefore, within our 12 samples that we loaded within our Gridion process, we only had 7 and 8 that showed higher numbers within our total base pairs within the million base pairs that were being shown, out of 59.76 for our barcode 7, 43.08 for our barcode 8. And within the total reads within the thousands was shown that 298.83 was being shown for sample 7, 215.52 was being found in barcode 8. That's the next one. Or one more, can I go one more back, please? I would like to also point out that for our sample 7 and also our sample 8, that the windmills that were being shown was for one more down. Go down. Was being shown there. The sample 7 and 8 that were being shown was for the windmills 511 and then for 511-2.
The both are.
Yeah. For our conclusion, a total of 125 samples were collected from the windmills in the Western and Central Agency within the Navajo Nation. A total of 39 water samples tested positive for collier horns, 51 tested positive for Pseudobunna species, 46 tested positive for enterococci, and 22 tested positive for Legionella species when analyzed using our IDEX tray. So none of our samples came out positive for the Helicobacter pylori cultures, but out of 125 water samples, a total of 11, the well numbers of 507-1, 507-W6, 507-11, 508, 509-2, 510-2, 511-2, 512-5, 8A 121, 9T 582, HK 402 came positive for Helicobacter pylori DNA using our PCR process. But these base pairs range between 8 to 1,000 base pairs, or approximately 92 nanograms per 5 microliters DNA max. But none of them showed positive signs for CAG A or BAC-A genes. So Helicobacter pylori base pairs within our concentrated water samples using our Oxford nanopore Gridion resulted in, of course, the sample size 7 and 8, which are windmills 511 and 511-2. For Helicobacter pylori genomes for a quantity to be read at 298.83 and also 59.76 million bases and then 215.52 or 43.08 million base pairs. Overall, the under-regulated water resources are considered as possible sources of spreading this Helicobacter pylori infection. There are studies that increase the H. pylori infection and associated gastric disorder in the Navajo communities, especially Western Agency. However, no studies on the possible resources, particularly the prevalence of this bacterium, is unregulated within the water resources within the Navajo Nation. Additionally, there are multiple studies on this chemical contamination in the unregulated water resources and very few studies on the microbial contaminations. But again, we focus on the prevalence of this Helicobacter pylori in the unregulated water located on the Navajo reservation. Through our study, we could not get any concrete proof of tested water resources being contaminated with this H. pylori bacterium, so we should not ignore the numbers of environmental factors that could interfere with the growth and survival lengths of this bacterium in the water resources. The current route examinations of the microbial drinking water quality. I can't really see that one. Kind of range. Can't really see on that part. Yeah, it's that one. Mainly detected conventional fecal indicators, but water-based non-conventional pathogens like H. pylori. That's why we use the Collie alert that would detect fecal matter within the sample size, within our sample water samples. That would show color forms. By the regular prevalence checks and tracking down the levels of microbial contaminations of any possible CAG-A or BAC-A positive H. pylori, in particular the water resource, we can simply design new strategies to achieve normal lifespans, to improve life quality, and to reduce the rate of morbidity in possible affected communities. This study is the baseline information to continue with more detailed strategies to isolate and identify the H. pylori and other pathogenic bacterium within the water resources, and then disinfect them accordingly. Helicobacter pylori can resist in high chlorine concentrations, so this also contributes to in what amount is it safe enough to increase the chlorine within the drinking water for it to be consumed by us. Moreover, to educate the local community on the proper storage and the consumption of the water and the possible contaminations in the unregulated water resources. Again, we need more detail and to regularly monitor and find the sources from where our community members are getting this infection. Thank you. I would also like to acknowledge greatly acknowledge my thesis supervisor, Dr. Shazia Hakim. I would like to thank my graduate committee members, Dr. Miranda Haskins, Dr. Fernanda Monroy, and also Dr. Nadine. And also our Tuba City Center Director, Ms. Phyllis Begay, Dr. Donald K. Robertson, who is our former MS Biology grant, Ms. Selena Yazzie for our grant manager, Allison Addison for our Learning Center coordinator from the Tuba City Center, Ms. Priscilla for our office coordinator, Cody from Tuba City, and also my laboratory members of Carlene, Maya, Vanessa, Caitlin, Harrison, and also Joyce and Paige, Molly, Romeo, and Patty. And of course, I would love to thank my parents, like my father, and then my family for the support and encouragement that they helped me from getting from here to this location weekly in order to conduct my thesis work. Is there anything else after that?
Thank you very much
for
for questions.
Sorry. The next slide will be for questions. So thank you very much. And thank you for your pretty much all your time and time. Thank you.
Say thank you to Anita also.
Anita. Where's Anita? Oh, sorry. Also, thank you very much, Anita. Thank you for
joining here. Yeah.
And thank you for giving me your time and upon my thesis defense. Thank you. Sorry, just my brace got tightened, and then that's why I had to really pronounce the wording. Just my braces.
Thank you so very much, Ian. So now Ian is ready to answer the questions. If any one of you have any questions for him, I saw so many of you from around who are joining us. Even one of the member, Gerlin Yazi, joining us from North Dakota. So that is something that is really very important for us to see that how much interest is there in knowing that how our students are working and trying to look for the problems that we have here and try to look for the solutions that how we can help resolve some of the basic issues here. And typically with reference to this Helicobacter pylori, I would like to mention here before we start having questions with Ian that there are many studies here, and we are continuously, especially with reference to Western agency here in the Navajo Nation, we are seeing that a number of cases are positive, particularly with reference to the high risk in the number of gastric and ulcers and gastric and stomach cancers. And mostly we associate this with the presence of Helicobacter pylori. We all know that it is one of the big source is contaminated food and water, but are we getting this really from the water resources here and how they are contributing? So this was one of the effort that he tried to do to look for some of the wells here that are unregulated and are being utilized by the local communities very regularly. So this is what the background of this study, and we are hoping to continue this work for more wells and more regularly with the different time contrasts, so that way we can see that if there is any difference that we are getting in our results if we are collecting the samples in different seasons. So this is something that we are going to continue. And now over to Ian. So please feel free to have any question that you might ask or any comment or anything that you would like to share here with to support and to make us available that Ian can answer here. And especially I want to thank our Diné College President, Dr. Deborah Dennison, who is here to support us and our students. This is really very important for us that how our administration is willing to support our all activities that we are trying to do in terms of promoting our students towards research and science. I also see here give me a second. So I see here Anita here is here from NAU. Is Dr. Anita is basically is part of our collaborating team from NAU under MITSI program, and Ian was able to take one of that class during the last semester also. So thank you, Anita, for being here. This is really very important again that how we are trying to develop the collaborations around to look for that how the things can make better. And before I get into some other things, I also really want to thank our both of our external examiners who are here, Dr. Miranda Husky, who provided guidance always to our MS students in terms of like providing the background with reference to the Navajo culture and traditional. And not only that, but also her expertise in a lot of other things help us understand the matters more in detail. Dr. Fernando Monroy is a name for the Helicobacter research from here in the Southwest, particularly from NAU. He has number of papers. He has done a lot of work on the gastric biopsies related to the Helicobacter pylori existence. So his presence, his guidance is also very important for us. And thank you both of you for your support and your help regarding this. And beside that, definitely like I saw here Dr. Give me a second. There is a whole list of individuals who are here, and I really want to thank. I saw Dr. Kamanilita, also chief, who also have done some work related to the bacterial and viral isolates from different water resources here in the Navajo Nation. So every one of you, your presence is really matter for us. That how we can continue to work in this direction for more efforts that we can do. All of our students here from our MS biology program, I see many of us are here. Some are here in the Tuba City campus. Some are attending from online also because they are currently located at different locations within the Navajo Nation. So yes, your presence, your support is very important, and it is also a matter of guidance for you that how you are working towards completion of your research and how you are going to moving forward. So let's see what you have for Ian and what Ian can answer. So please feel free to have any comments or any suggestions. Thank you very much for being here to support us.
I don't have to ask any questions.
Any question or
Or
any comment?
everything was in the PowerPoint.
Hi, Ian. I'm going to be the first one. Can you hear
Anyone
me?
has any question? Or
Can you hear me, Ian?
I think it's the same difficulties from the last.
let me see if I can.
Ian, can you hear me?
Oh, sorry.
Yes.
Of course, Dr. Monroe, I can hear you now.
Okay. Well, great presentation, and I'm a little bit surprised that you also got a low prevalence. As you know, I think sometimes the storage after collection of the water can cause the organism to transform from the spiral to the round that is called the cocoid form. But when you look at the DNA, I think I was very pleasantly surprised that you were able to identify some of those, meaning that we are heading in the right direction, because if 64% prevalence is what is known in the Navajo Nation, that organism has to come from somewhere. And maybe a little bit of tweaking with some of the collection, or maybe, I don't know, do something with the samples that bacterial DNA from other species might overwhelm H. pylori, and then the sensitivity is not there. But I was pleasantly surprised that you were able to find them, because we couldn't. And even if it is a small number, that's heading in the right direction. I wanted to congratulate you for that.
Well, I'm pretty sure you would have did a better job than me, Dr. Monroe. I pretty much learned the techniques from when I visited at NAU campus. But one thing I would like to add in was that we did two different types of tests. We did one with the DNA extraction, and then the concentration DNA extraction, and then we did one with and without the PCR. And we got better results when we did it with the PCR before running it on within our Gridion sequence to be read.
Perfect. Great job.
Well, thank you very much, Dr.
Thank you.
Monroe. I appreciate everything that you've done for me.
Okay, Ian, since nobody wants to ask question, can I ask you another question?
Sure, sir, of course.
And this is a question that I ask all my grad students or students that I serve in their grad committee. And it's a very basic question, but the question is, Ian, if you had to do this experiment, this project over, will you do it the same way? What would you change?
One thing I probably would change is probably take two different timelines, one done in the summertime and then one done in the winter time, just to check and time see if the temperature would also make a difference within this type of experiment. Another thing I would like to do would be to either repeat the process, because I know that it requires the 5,000 at a 10 minutes, 5,000 RPMs. But for us, we have for our centrifuge, we had to run it for 20 minutes, like twice the time. And then also pretty much being more fragile and also being more careful, such as being in sterilizations, such as the use of ethanol that we were able to use when working within the biofilm, but within the biohoods. But another thing would also be to work with, or before doing anything, would to just do a continuous procedure in each, such as taking the water samples in the first day, bringing them back, doing the concentrations, and then afterwards doing our doing our DNA extraction in order so that we can get the lysate, and then pretty much checking the quantifications, making sure they meet that 20 nanograms. And then next, just being able to run it through our PCRs within our PCR conditions, for that they can be run through our Gridion, which will be our devices. And I would
That
love.
is perfect. Stop there. All I wanted was, because remember, whenever we run a project or we run an experiment, at the end, we always realize we could have done it in many different ways. If you had replied, "Oh no, I wouldn't have changed anything. It was perfect," I wouldn't pass you. I said, because that meant that you didn't learn. But you learn a lot because you are talking about so many variables that you can implement. And if you had to do it again, what you change, and that is meaningful. That means that this project served you a lot, which is teach you how to set up a specific question and how to go out there and accomplish that. That's all I wanted. Thank you.
Thank you very much, Dr. Monroy, and thank you for your paper that was published in 2022. It was like a great help to our project and for most likely future projects in the future.
You're welcome.
Thank you.
I had a question.
Yes.
Congratulations, Ian and Jana Inla. This is a really informative study and very important to our Diné people here on the Navajo Nation. My question for you was, given the fact that many Diné people continue to utilize these unregulated water sources across Navajo, at the end, you made a recommendation that you would provide ways to achieve proper use, storage, and consumption. So I'm wondering if there's one recommendation that you could make in the immediacy so that people know how to achieve that proper storage to reduce the incidence of H. pylori infection. Thank you.
Yes, thank you very much, Dr. Haskins. Well, pretty much when it comes to these, of course, we live within a rural location in the terrains, and then traveling to these unregulated water resources. There's like so many thousands of windmills across the Navajo Nation, and to obtain these water samples and to bring it back home, it takes a lot of time and a lot of gas that not a lot of families have. But one thing is to check the storages in which they're being consumed and also around them, so that it's not being kept within unsanitized locations. Also to also go into the nearest facilities and also be getting analyzed and diagnosed to check if there's any prevalence of this bacterium that could be within a household, since within a family of the Navajo reservation, there's more than just 5. There can be even 10 into one household. So therefore, the person-to-person contact is really close. Therefore, this bacterium can be able to infect others.
I have a question. Congratulations, Ian, on completing your master's program. What do you plan on doing after?
After would mainly be trying to obtain my MLT certification so that I can apply for occupations within the laboratory setting to help reduce the strain on also the amounts of reads in laboratory, also within the laboratory setting, and also to increase production. That's what I want to do is either work as an MLT or medical laboratory scientist to use my master's degree and also my bachelor's degree for these types of settings within the laboratory, and to transfer the same laboratory skills towards these areas.
Any perspectives on teaching positions?
My mom was interested, but I don't know. We'll see. If I can get the patients down, which I've done with the project, then I don't mind leaning towards that. It would be something that would be greatly a good interest of mine.
Thank you very much. Dr. Carmenita, Dr. Chief, please.
Hi, congratulations, Ian. That was a really fascinating presentation. Dr. Hakim, I just want to clarify that I think you're talking about Dr. Carletta Chief, although we've worked together in the past. I'm Carmelita Chief. I'm a PhD student at NAU, and I've worked with Dr. Monroy and the Navajo Healthy Stomach Project. So definitely a very interesting topic. Ian, so my question really revolves around reflexivity and the practice of reflexivity as a researcher, which is just basically, reflecting on how you as a person and your background, your beliefs, all of that, your perspectives, how does that influence your work on this project? I just wanted to get a sense of how maybe going through this has changed you, how your background just kind of informed the approaches that you took with your work. I'm just interested with that.
Before starting the master's program, I started a program with Workforce, which was I was associated with the MPL, which they do checking on unregulated windmills and then repairments, and also the fence repairs, and then also they do cattle guard repairs. Cattle guard repairs. So I kind of got the sense of majority of where all these windmills were located. So within the Western Agency, because I was able to travel to majority of these windmills, even in rural locations, some of them were damaged. And then some of them had minor repairs, such as when on a windy day, you could even hit majority of the pipe, some of the main line, and you were able to release the rocks in order for the water to continue pumping. And around these unregulated water resources, such as the windmills, I noticed a lot of defecation, such as from the livestock. And it kind of got me thinking about when starting this project upon if these types of feces were also one of the contributes towards the contamination of the water resources. Another would also be when we also haul water for our livestock. So therefore, I kind of got the sense of how this type of work would also contribute towards people that use these unregulated water samples for unregulated water samples for their crops or their livestock or even their personal use, since not all the reservations have, not everyone on the reservations have running water. And of course, my dad, he does silversmithing work, so the work kind of transferred between here and the lab. Just being able to work with small items and then having to work in the lab was the same type of techniques. So that's where I kind of got the sense of using the pipettes and or having to work with small samples such as the primers and also the calculations and such as measurements that are being done. So it's just like the transfer of either what I've been through an occupation, and then also what I see around me, and then also the techniques that I grew up with.
Awesome. Thank you so much for sharing a bit more insight on that. And thank you again. And Like I mentioned, I'm a PhD student. I'm also going to be presenting on work that I focused on around the topic, but more in the realm of health communication. So definitely going to add you as a reference for the two presentations I have this week. So you're already going to be cited.
Well, thank you. Thank you very much. And of course, you're going to do a wonderful job. And I
Thank
hope
you.
all goes well for you.
Thank you very much. Yes, Carmelita, I was not mixing with Dr. Chief, but I have seen some of your work that you have published, so I was talking with that reference. So thank you for being here. It is really very helpful. I see Mr. Gary from ONSFA. ONSFA has remained very big support for our research students. So thank you very much for being here also. And any more questions? Any other questions, please?
That's it. That's okay. They can color it then.
Dr. Dennison, would you like to say something?
No response. I think we're good.
And here is a question from Octavia about that. Did you test any regulated samples like tap water or as a comparison to other unregulated samples?
No, we did no tests on the regulated samples. Just majority of them were all done on unregulated water resources, such as windmills, water springs, and so forth.
Thank you very much. Anita, any comments, suggestion, anything for future?
I'll just say congratulations. That was a wonderful presentation and I learned a lot and getting to know you and getting to hear you speak. And I hope the next part of your defense goes beautifully. I know it will.
Thank you very much.
Thank you very much. I think Joyce asked a question.
Okay.
It's in the comments, I think. I didn't really get to read it.
No, there are comments, congratulations comments from Mr. Benali, Mr. Sheldon Benali and others. So thank you very much. So I think that if we do not have any more questions, it is almost 1:01. We have taken whole 1 hour of you. So thank you very much for spending your lunchtime with us and supporting Ian's work. Again, it is not possible without so many of you who provides guidance, who provides support, who provide suggestions and who help us to improve more. And there is always a possibility of improving more and more. So please feel free to provide us your feedback and we will be really try to incorporate all those suggestions and your feedback for our future programs. And hopefully Ian will be interested in doing some more higher studies or looking for PhD program also. So again, thank you very much and have a good day. Bye. Thank you very much.
How we can eat. I was like, no.